last time. Okay. Right, enable content. And make a start. Okay, so this is your pre-lab for tomorrow's lab. So we have the same approach that we've always had for each of these groupings. But the first one I want to start with is actually a small video because we did Hymenoptera last week and we're doing flies this week to give you a bit of an idea. So we'll start with this one.
Fly versus bee.
I am bee.
I'm fly.
I collect nectar and pollen from flowers and bring it to my community for nourishment.
Oh, I eat dog shit.
I keep my young safe in a nursery until they mature and are ready to join the community.
Oh, well, I just dump my kids in a garbage can.
People are afraid of me because I've got a nifty weapon and I'm not afraid to use it.
Oh, people just hate me because I'm gross.
I spend the whole day doing chores for my community.
Well, I just fly around, piss people off, you know. Yeah.
Okay, so you're getting a bit of an idea about the difference between Hymenoptera and Diptera. So bad reputation if you're a fly, and we'll go into some of those reputations and things. So earliest fossils, Upper Triassic, 225 million years ago. Okay, so if we have a look at that to begin with, here is our representation, and we can see flies here. So we're getting earlier origin point than both the Hymenoptera and the Lepidoptera, similar to where the Coleoptera and the Hemiptera were quite early on, because as we talk about in this lecture, flies were probably associated with scavenging and floors and litters and things like that. So there was plenty of those resources around. As we spoke about with Hemiptera, there was gymnosperms and mouth parts could access them. So we've got flies coming in pretty early, but we've always got this point when the angiosperms begin to dominate where they start to radiate even further. So amongst the three earlier of the five that we've looked at. Number of species, 150,000 described species, but we'd say probably twice that, 300,000. If we compare that again to what we've looked at in the previous lectures, remember that Coleoptera are around 390,000, so they're way out there. The leps are about 160, so the next most speciose, then the flies about 150, the wasps about 115, and the Hemiptera are about 110. So up there in that middle bracket with large numbers of described species amongst the big five. If you want to go below that, the next most speciose order is the Orthoptera, and there's only 20,000 in that. So it's a big jump, which is why we spend most of our time with the five major ones and not with those. So we did a little bit of Orthoptera in the lab, if you remember. 130 families in the world, 98 in Australia, and 8,000 species of fly. In Australia, 80 families in Tasmania, so a lot of families are present here. And again, if we think about mammals in the world, only 5,000 species of mammals, so we've got more species of fly than we have mammals in the world, and I think reptiles are about 8 around the world. So again, you get appreciation of just how enormously diverse some of these insect orders are. When we look at it in terms of phylogeny, let me see, there it is, Diptera right there. So it's close to the scorpion flies and the fleas, and also has a probable relationship with the Strepsiptera or twisted wings. Remember when we did the Coleoptera lecture that there was morphological evidence that it was more related to Coleoptera, but molecular evidence that it's probably more related to Diptera. So verdict's still a little bit out depending on who you want to trust and believe. Okay, let's have a look at flies in general public arena with the introduction first. So this is a pier that I visited in Liverpool, which has the typical sort of approach to flies, gruesome, ghouly, vomiting up green slime down there. So in your horror train ride, that's what you'd want to get into. Flies, great cartoons for flies too. If you're a fly, you want to go travel to dumps around the world, not nice fancy places. And in Australia, flies have lots of reputations. Bush flies being one. but also mosquitoes. So here we've got a postcard that I got from Queensland, where it's insinuating the beer's good and the locals are really friendly, but you can see this is in sort of 4X colours, and you can see, and I've told you already, that flies taste with their feet. So they got socks on, so they can't taste, which is why he's drinking that Queensland beer. Okay, well, I better not go there first. In terms of flies, remember when we went to the first lecture and we looked at what was underrepresented? People didn't eat flies as much as they overate Orthoptera and other things. However, if you're a German backpacker, could make you survive. So this guy, a couple of years ago, was found after 10 days out in the outback, and his claim was that for 10 days he ate flies. He got enough protein off eating flies. So a lot of flies out there that helped him survive. So generally not, but German backpackers maybe. Okay, in terms of commercials, this is a classic in Australian commercials, Louis the Fly, which I'll show you the original one of before. I worked in a lab in Sydney for a while that housed Louis the Fly. Because whenever they did their Mortein commercials, they used to come in and collect lots of flies off us. So that was the home of Louis. But you can see here, flies spreading disease, wiping feet on your food. These are old posters. And the kids' book here with the noble knight on a horse with its lance fighting off a giant fly coming out there. In that case, he's been shrunk, I guess, because it's coming out of the bin. Let's have a look at the original Louis the Fly. You may not have ever seen these.
Louis the Fly, on Louis the Fly, straight from rubbish tip to you, spreading disease with the greatest of ease, straight from rubbish tip to you. I'm bad and mean and mighty unclean, afraid of no one except the man with the can of Mortein. Hate that word, Mortein.
One spray and Louis the Fly, apple of his old mother's eye, was Louis. Poor dead Louis, Louis the Fly, a victim of Mortein. Mortein.
Mortein is the safest.
Okay, we won't tell us what Mortein is. So that often it's one of those words in Australia, a bit like Esky, that's a product name, but Mortein often is referred to for fly sprays. It was actually developed here in Australia around the time of that bushfly dung beetle program. So another one here for flies. This is my last commercial, so this is it for the year.
I was born 24 hours ago. I wasted my life. Don't be like me.
You never know what the product is till you get to the end of these commercials, do you? So there we go. Okay, so yeah, make most of life. And there is another saying, I wonder what the fly on the wall heard, and there's a couple of examples of that. This one is an ad for Merrill Lynch saying, you know, world's largest investment company, what no one overheard. That occurred just before the global financial crisis, so it probably heard things about what was going on in that company that most people would be curious about. This one is a witness statement, and for my final witness I call to the stand the fly on the wall, and of course there would be one at the bar too, so I am the fly on the wall you speak of, what would you like to know? So it knows everything, so the all-knowing fly. Okay, so let's now try and find out what a fly is, rather than just a fly on the wall or a commercial with the Bacardi. Okay, I'm not going to highlight this one because you all know this now. Diptera, one pair of wings on the mesothorax, so that one should be really clear to you by now. Hind wings reduced to club-like ball tears, and we've said, and we'll come back to this again. Here you can see on the promesometathorax here, we can see the hall tears, which look like little chicken drumsticks, and these sort of things are gyroscopes for orientation and things, and we've already spoken about how fantastic the two-wing option is for flight and flight speed and all sorts of things when we did the lecture on flight. Okay, if you're an adult, you've got a number of variations for mouth parts, but you're not going to have chewing mouth parts. So no adult fly is going to be chewing up anything, okay? It's going to be sucking, cutting, sponging, or sponge-like mouth parts. So we'll talk about, we'll show some of those, but they all relate to some level of liquid diet as an adult. The other thing is we have complete metamorphosis, okay, so the holometabolous, the larvae are legless. They have chewing mouth parts or mouth hook-like mouth parts, and we'll show those in a minute. And they often pupate within a hardened outer case, which is called the puparium. So in this case, I'll show you with slides a bit about the puparium rather than use this as the example. Okay, so let's have a look at those mouth parts that are mentioned there on that list. And here we have here larvae that are chewing mouth parts or mouth parts. So here is mosquito larvae, and you can see here mosquito larvae with a strongly sclerotized head capsule, and also chewing, filter feeding sort of mouth parts here in this case. So we can get chewing mouth parts. And this is the other option that we have for the fly larvae, which is like a hook-like mouth part, which you can see here on the end of this. So there isn't that much on it. If we look at this in detail here, this is known as the cephalopharyngeal, so it's surrounding the head. And you can see the mouth hook. So if you drop a maggot into something like potassium hydroxide, it melts away. Then you can look at these sort of cuticular teeth and try and get some identification on what that maggot might be. But in forensics and all these sort of things, it's a hell of a lot easier to try and rear it through and get an adult fly, because there are a lot more features on an adult fly, or to get molecular techniques going to identify larval flies. So there are problems with that. But certainly, that's the two sorts of mouth parts. However, I do realise I now have one more video I've embedded in here last night. I have an alternate mouth part as shown in this science fiction.
Giant maggots?
No. Doctor, it's exactly your cup of tea. This fella's bright green, apparently, and dead.
It's time that the world awoke to the alarm bell commission.
Giant maggots indeed. Devilish creatures, small but filthy byproducts of your technology.
What kind of insects are they going to turn into?
Do you read me? Hello? Hello?
Joe, are you there? That mine must not be investigated.
Oh, I missed it. Sorry.
No chance.
We've got no chance. There, look. You missed it. Sometimes it doesn't go back, but it's got teeth and everything, that maggot. I don't think there's any more examples of it. Does this go back to the beginning? Yeah, that's the problem, because I can't advance this thing under these. So anyway, it's got teeth. It's Dr. Who. Green Death it was called. Okay, also in the lab today, here's another interesting larval form of a Diptera. This is in the family Surfidae. The hoverfly is a genus known as Microdon. We've got some in the lab tomorrow, which I collected. These actually live in ant nests. And for a long time they were thought to be mollusks or slugs, snails. They just don't look anything like it. So you get some pretty interesting body forms, and you can see them here in the ant nest. That's what the adult looks like. And students do come up with Microdon. I think we've got a few in the collection, adults, but I've only ever found the one example larvae. And these will be found actually, if you're looking for them, they'll be hanging around certain species of ants' nests. They're not the flowers and things, they're hanging around waiting to get into the ant's nest and trying to drop their larvae into there. So, yeah, Microdon. Okay, this situation around pupation and the puparium. The puparium, which is restricted to a certain grouping, and we'll talk about that in a moment in the flies, is actually the hardened larval skin that it pupates within. So the actual larval skin hardens and it pupates within that, and the puparium looks like this. It looks sort of like a rat dropping, okay? And it's only in a certain grouping of flies, and we'll go to that when we go through the next few slides. Okay, so we have two suborders in the flies, Nematocera and Brachycera. So Nematocera, some key things that we can look at. Firstly, their antennae are longer than their head, and often their thorax greater than 6 segments. So there's quite long antennae in these. They're gnat-like, so they often have very long legs, and a maxillary pout that is 3 to 5 segmented, which is down here, which we can't see on this particular example. I think maybe I can show you that. Often nocturnal and swarming. I'll come back to this slide in a minute, but I want to show you those features to begin with. So here we have the antenna, longer than the head, and it's got quite a number of different segments on that antenna. Here's the maxillary pout. We said that would be 3 to 5 segments, and we can see here 1, 2, 3, 4 segments. So there, and we've got the long legs. So those things all put it in the Nematocera grouping. Okay. The next one is the head capsule, which we did look at in a slide or two back. So the head capsule is sclerotized, so it has an obvious head capsule like the mosquito larvae, which you can see here with the mandibles moving horizontally. But the next one I want to talk about is a terminology that we haven't really addressed, but is useful to address when we get to flies, but it does exist for a lot of insect orders. So we can have pupae that are either obtect, where the legs are externally fixed. So this is an obtect pupae here, and you can see here the pupae. So they're all tucked in into the pupal casing, but they're visible from there, but they're not free out there wandering around like this. So that's an obtect pupae. So we either get obtect pupae, or in some cases in these groups we get exorate pupae. In this case you can see here where the legs are actually free, not enclosed and squashed up like they are in obtect pupae. We'll see these things. I mean, obviously a honeybee pupae is exorate, so it does exist in others as well, but in this Nematocera we're either seeing obtect or some exorate pupae, no puparium. Okay, the Brachycera, these are stouter flies than Nematocera. We talked about Nematocera being gnat-like. They have maxillary pouts that are much more reduced, only 1 to 2 segmented, and their antennae are mostly aristate. So this is that aristate antenna that we looked at and asked about in the lab, where you can see that little whip-like aristate antenna off the other antennal segment there. And they have something known as tallinal fissure on them, and we'll talk about that and show you that soon. Okay, then we get to the larvae. No vestigial head capsule, or vestigial head capsule, very little, small head capsule. Usually it's just this hook here, and the mandibles and that hook move vertically unless you're in Doctor Who. Pupae can be obtect or coarctate as well, but they form a puparium. So let's have a look here. And this is a puparium here, coarctate or puparium. So here are our groups, Nematocera, and here's the Brachycera. And within all these Brachycera here, we have the obtect pupae, or in some cases the exorate version of the pupae. But here within this grouping, known as the Schizophora, within the Brachycera, all of these form a puparium, which remember is the outer larval skin of the third instar as it pupates. So if you were to find this, you'd know exactly what it is, because it's the only thing only exists in Diptera. So 40% of all flies have a puparium, and 78 of about 130 families have a puparium. So that's a straight giveaway what sort of fly that you'd be looking for. The other thing is that I said it has a tallinal fissure, and this is a problem for flies to get out of their puparium. So it only occurs with flies that have a puparium, that they have a weak point on their pupae, but they don't have chewing mouth parts, so they can't chew their way out like a wasp can out of its pupae. So they actually inflate their head up. They do a party trick and get a bigger and bigger head to actually push themselves out of the weak point on the puparium. So here you can see it's just come out, inflated its head, and then moves its way out of the puparia. So it creates something known as a talinal fissure, which is this point, structure around here. You can see on the front of the head where the head would have been blown up to get out of the puparium. So if you see a fly with this sort of structure, its talinal fissure, you'd automatically know that it would be one with the puparium, and a little bit more about its larval life cycle might be understood, and we'll talk about that in a minute. So here we have a beautiful shot of one of such of these, and you can see here the talinal fissure around here on this fly. And there you can see beautiful aristate antenna, etc. Okay, let's have a look at general biology and start with larvae first. And generally, in the Nematocera, the larvae are associated with moist habitats, which often is aquatic habitats, okay? So this is a black fly larvae here, you can see here with its filter feeding apparatus. So the majority of Nematocera are in moist habitats, often aquatic. Brachycera have managed to radiate out into drier habitats, and their hosts that they can feed on or live in could be plant tissue, they could be predators, they could be parasites, or myiasis, which is feeding on living flesh, as you can see here with a sheep that's been struck by a sheep blowfly. So here we can see a Brachycera, typical Brachycera, with just that vertical moving mouth hook, and here's their pupae, their spiracles on the end here with the puparium, the different colours as they get older. So they've moved out into drier habitats with that puparium and the ability to withstand more drier areas. Okay, adults. Now remember we said really they only take liquid food, so they've only got sort of a sucking, sponging, cutting mouth part. So some sorts of food include here yellow dung flies all over dung. We can see here taking out some sort of sugary solution, here feeding off another dead insect, and here taking nectar from flowers. So we're looking at liquid food, so we could never be accusing an adult fly of chewing damage on any sort of plant. It's not going to happen. However, quite a lot of flies have decided, or some significant groups of flies have decided, that the liquid diet they're going to take is a blood diet. Okay, and this is where the flies come into their own in terms of their importance, and we'll talk about that as we move on. But we have three groups here: the biting midges, the black flies, and the group down there known as cycodids, or sandflies. In Australia we call them moth flies. All of these have a fairly short proboscis, so they're not plumbing very far down when they're biting the host. And if you've ever been to a place like Canada after the snow melt, you're just going to be harassed no end by things like black flies. So it's a very unpleasant environment when these things get around. And these sort of things, when they're hassling black flies, for instance, you can lose a hell of a lot of body weight on stock in those areas just from the harassment that these things are continually biting. And you can feel them biting, but they're small little things to see, so they're really harassing. If you want to get down a bit further, you're going to modify your mouth parts even more. So you've got that sucking mouth part that we looked at of the mosquito, so you can get down to capillaries and things like that. So we have the labium sheath, and in that we have a hypopharynx, the mandibles and the maxilla, all sheathed in there with 6 stylets in order to take blood. The other one that we did speak about when we looked, there's another one to come, was a cutting, sponging mouth part, which was seen in things like horseflies, tabanids, march flies, where they actually have modified their mandibles and their maxilla to be cutting, and they rasp on the surface and then suck it up with their labella, the blood that rises from that. That's why they are very easy to feel when they're biting you, as I said in that original lecture that we did on that. But also, again, are quite an issue for livestock who can't necessarily swap them. Sponging mouth part here is the housefly. You can see that labellum just there taking up that liquid quite nicely. Okay, let's have a look at the economic importance of flies. and where I've drawn the notes here. And the first thing I want to say is that compared to all other orders, Diptera are most significant in the medical and veterinary space. Okay, so that's where they really stand out in terms of economic issues. And here's an example of a slide that was out not that long ago with the Bill Gates Foundation looking for the world's deadliest animals, and we're pretty terrified of sharks, but this is about 10 per year for people killed, but mosquitoes are responsible for vectoring diseases that are killing, getting closewards to 1 million people a year in some years. We're responsible for killing a lot of people as well, quite clearly, but the mosquitoes are the number one out there in terms of that. Now let's have a look at some significant human and veterinary diseases transmitted by Diptera. And we can see here mosquitoes are one of the big groups. So in this case we're talking all human diseases, but we've got malaria, yellow fever, dengue fever, encephalitis, Ross River virus, filariasis, Zika virus, which was around at the Rio Olympics time, Barmah forest virus, which was only detected up on the east coast of Tasmania 2 weeks ago. So there are a lot of viruses, particularly transmitted by mosquitoes. Then we have one of our favourite insects, the tsetse fly, which had that amazing birth where it only gave birth to a larvae and it's about to pupate. But that transmits sleeping sickness and nagana, which is a disease for livestock, protozoans. Tabanids or horseflies can be responsible for transmitting anthrax and a couple of other diseases. Sandflies, leishmaniasis, river blindness by black flies, and biting midges, blue tongue virus, and a few other types of filariasis. So you can see here a number of different pathogens, viruses, protozoans, bacteria, nematodes, it varies, and a number of different hosts and regions. Typical tropical regions is the major thing for these. Okay, let's just talk about transmission of such diseases and a couple of points that I want to make in terms of that. The first option for transmitting diseases with flies, and we did, I think, mention this a little bit with Diptera as well, is that you can be a mechanical vector, which means that you pick it up on some part of your body, and for the period of time that it's externally alive on your body, you can transmit it to other areas. Okay, it's not actually replicating or living inside you at all. So a housefly is our best example of a mechanical vector, and things like typhoid, cholera, dysentery can be vectored around through things like houseflies. But the important point with mechanical vectors, this is not the only way that it can be transported. People not washing their hands and salads and all sorts of things can give you dysentery, etc. So there are other routes for that to get through. But houseflies are a mechanical vector. Now we have the other option, which is a biological vector. And that is where it actually replicates inside the host, and I've got a couple of slides after that to illustrate it, but we'll do the tsetse fly as our example here, and we spoke about sleeping sickness through Africa. So minimum of 20,000 new cases a year and 50 million people are at risk. So this is the band where tsetse fly is present and where people are exposed to sleeping sickness. Similarly, nagana is a similar disease to sleeping sickness, but it's in cattle. And here's your tsetse infected areas, and here's the areas that you can actually raise cattle. So it completely excludes cattle raising from parts of Africa because of tsetse fly. So large regions, which then has malnutrition issues for people that can't rear cattle and livestock in those areas. So can have quite profound implications not only in human populations but in livestock issues. So in biological transmissions, the only route is via that vector, the insect. It replicates in it. So if you want to control something with a biological vector, then yes, elimination of that vector could get rid of the disease. Okay. Once it's been replicated inside the vector, it's going to be there for life, and it's a property of the disease, not of the vector. So you could have some species of insect, such as a horsefly, that can transmit both mechanical and biological. It's not a property of the vector itself, it's a property of the disease. And the other thing that's important when we're thinking about veterinary and medical diseases with flies and other things, and similar, I think Calum will come across this a little bit with your viruses and your pests, is there can be single cycle or a reservoir host cycle. So a single cycle is just pathogen human insect vectors, so that's malaria. So, you know, the mosquito picks up malaria from a person, feeds on them, goes out, digests that blood meal, then comes and feeds on another person. Then the malaria comes through the saliva into that person, and then that's how we get the transmission of malaria from a biting mosquito from person to person. So you break human vector contact, there's one way of getting rid of it, so bed nets, eradicating the vector, or chemotherapy like we do with malarial tablets. So there are a couple of approaches to doing that when it's a more simple approach. I'm not saying malaria is simple because it's the worst disease that we have to deal with in tropical areas. But if we have a reservoir host, it makes it really complicated. So with yellow fever and with Ross River virus, the reservoir host is macropods, kangaroos, wallabies, those sort of things, and possibly also some other marsupial species. So there are two cycles there. There's the reservoir host, the pathogen in the vector, and humans, pathogen in the vector. So control of that, if you try and do something in the human population, make bugger all difference because there's a reservoir host out there, okay? So you've got to separate the reservoir host from the humans. That's why we get things like, well, they're not fly related, but I'm trying to think of that disease in Africa that they try and keep people away from monkeys. But anyway, it's similar here with Sylvan yellow fever. It's the same thing with this, that there's a route where the natural incidence of disease is in the reservoir host, the monkey, in the rainforest, but then people come and plant bananas closer and closer to rainforest regions and get in contact with those mosquitoes. Those mosquitoes spill over and feed on the humans. It's not what their normal cycle is, but they do, and then humans bring that into the population, and we get populations of yellow fever going through there. So it's difficult to control, much more difficult control when there are reservoir hosts. Okay, let's have a look at a few other things in terms of economic importance of flies. Myiasis, feeding on living flesh. This one is the sheep blowfly. On your handout there, I have given you something on sheep blowfly. In the last few slides, I'll also talk about sheep blowfly. So basically, they're feeding on a live host. It's a pretty nasty sort of thing, obviously, to be fed on alive. A bit like the botfly that we showed, where it's feeding on people while they're alive. Botfly's right here. So that is the botfly. If you stayed and watched that over there, we can see the botfly living in the person. But we also have some that are external parasites. This one is the ked or louse fly, which you find on sheep. And they have a very reduced wing, but they're hanging off sheep and feeding on sheep there. So quite an interesting looking fly family there. And then we've mentioned already, but the nuisance value of flies can be extreme. So buffalo fly, stable fly. In Australia are certainly places where these breed, and they breed as larvae in sort of moist environments and dung of certain ages, etc. So you'll get lots of these breeding in areas, then they'll start harassing stock, biting them with those cutting, sponging mouth parts and harassing them, and you'll get loss of stock weight through these sort of nuisance flies. Bushfly, again, we've already mentioned the bushfly and the issue of how the bushfly was very important to being able to now have al fresco dining with the dung beetle, but it obviously is a pretty big nuisance in a lot of areas still. Okay, if we're going to start to feed on plants, we really have two approaches. We're never going to see them chewing plant leaves, like you might have beetle larvae or something, but you do see leaf mining in flies. So there is one family, agromyzids there, that are leaf miners. So you can see these all moving through here in leaf mining. I think there might be some Hymenoptera that are leaf miners as well, so it's not guaranteed to be a fly, but we do have fly species that are certainly leaf miners. So it's one sort of damage we can get through flies and their association with plants. Another option, which we'll talk about in another slide or two, but I'll bring up now, is also they can form galls. There are some that form galls on plants, fly species, but remember what we've already said, in other lectures that galls could be formed by throops, they could be formed by Hymenoptera, they could be formed by Diptera, etc. But galls is another product of flies that could be damaging to your plant. But the real big doozy of fly issues, and we're very aware of this in Tasmania, is fruit flies. Diptera. Now we've got specimens of Queensland fruit fly in the lab for you to look at tomorrow. People really get confused what a fruit fly is, thinking all sorts of things are fruit flies, and we've already started to now understand what a fly larvae looks like as opposed to a caterpillar larvae, which is what typically people are seeing inside fruit. So this is the Queensland fruit fly, which we can see here. Very striking markings. So if you're on the east coast of Australia and down into Victoria, where populations seem to persist now, where some of our problems are coming from, then Queensland fruit fly is going to be a problem. If you live over in Western Australia, it's Mediterranean fruit fly, which looks different to this. And I have managed to push Mediterranean fruit fly at times out of some of the big growing regions like Carnarvon in the west through sterile insect technology. And at the moment there is a sterile insect program being funded in Australia with rearing facilities in South Australia to try and help with controlling spot outbreaks and things, so releasing sterile males to reduce populations. But if you're moving around on the mainland, and obviously also when you're in Tasmania, when the zones are on, there are zones that you're not allowed to bring fruit or anything through. We see that, of course, when we come into the airport here. So a huge issue, and there is sort of biologically eradicated and politically eradicated. It's a very important thing for quarantine not to have any possibility, or to dismiss any possibility that fruit flies could exist in an environment because of the export value without fruit flies. When I was in Los Angeles working over there, they also had Mediterranean fruit fly outbreaks, and the relative importance of that was such that they used to fly like sort of Vietnam, like bands of helicopters across with spray gear across urban areas to try and take out the fruit fly through those areas. And it caused a lot of stink because Americans and their love affair with their cars thought it was upsetting the Juco on their cars. Probably weren't so concerned about the health effects, but the Juco on their cars was very important. Fruit fly, big one. I'm trying to turn this around and also think about, well, you know, I've said pretty nasty things about flies and given you some pretty awful videos about flies and horror stories. So let's try and think some good things of flies. Okay, so there are good mosquitoes. Myxomatosis, which is spread, can be spread through mosquito movement from rabbit to rabbit. So here we have the rabbit wanting his insect repellent thanks to stop being bitten by mosquitoes. There is also a flea involved with other strains of these, but which is why it didn't do so well initially when the release was all about mosquitoes in drier environments, because there aren't a lot of mosquitoes in those populations or areas. So yeah, it can be transmit beneficial diseases. Okay, this is the warning. The next few slides are going to be a bit more gross. So think of it as cute little maggots. Okay, so this one here is maggot therapy. Okay, so with the current situation with antibiotics and things, more and more so that wounds and things are not being able to be properly healed and treated through medicines. So there is quite a return to maggots and maggot therapy. I've had quite a few phone calls over it, but you have to really have a proper sterile facility. I can go back to the cute little maggots if you like for a moment. Sterile facility there for rearing them. So the UK has gone into this in quite a big way because of their worry about resistance. So you would put a few little tiny maggots on there under the wound dressing, and they clean out all the dead flesh and also cancerous tissue. So they found this out originally in the Crimean War when soldiers were coming back who had maggot infestations on them when they're coming back off the battlefield, that their actual legs and amputations and things were a lot better from having maggots on them than not. And then that was confirmed in the First World War, with the horror of the First World War, where troops that died, well, that had wounds that were in shadow or shaded areas versus more exposed areas where you had flies or no flies, had differential survival rates and things of based on flies. So the flies are actually cleaning up a lot of things. So so they are quite a useful method, maggot therapy, for looking after wounds and cancerous as well. Okay, this one's not quite as gross, maybe, I hope, for you, but forensics. So again, this is a pig that we decomposed up here in Hobart for a police exercise time. And this is 6 days after that pig was put out there. So this is in the peak of summer. So within 6 days it looked like that. And you can see the maggots coming out of there. So the whole principle of forensic entomology is that the development of the larvae follows a certain pattern according to temperatures experienced. So when someone is killed and put out or exposed to an environment outside, then immediately the fly species will arrive. And then by the size of the maggots, then you can then time how long that person had been out there and exposed in the environment. You can also do a lot more with this as well, where things have been moved from location to location that it doesn't match, because you'll get certain fauna in shaded and certain fauna, say, in open conditions. So if there's been a subsequent movement of the body, there is also other methods that you can use. And the puparia are really good little things. So they're going to come out of there and they're going to move, you know, tens of centimetres to a metre away from the body. So if you're going and looking for something, you pick up the puparia and bring it back. And they're like, after these things have all got dry, they're a little gold nugget of information, because if there is any chemicals in the body or anything like that, you can actually analyse what's in the puparia and pick up what has been present in the body through drugs or other things like that, or poisons that might have been there. So the puparium are really useful as well. Okay, this one, Drosophila, often called fruit fly, but preferably called vinegar flies. Small little fly species will be in the lab tomorrow as well, so we're rearing them at the moment, but fantastic for genetics. A lot of the basic fundamentals of genetics and human genetics is there. Menzies has colonies of these things at the moment over there, looking at brain related issues and memory related issues, using vinegar flies as their models. But there is also used in models around nutrition, obesity, all sorts of things currently, where vinegar flies with their high turnover and genetic mutations are really able to help answer quite a few things. Pollination. Pollination is another big thing that flies are helpful with. They're not as fantastic as the Hymenoptera, which we said could be very dedicated to following, like a bee. Will go from that same species of flower to another same species of flower, and so on. These things, flies are great because there's a lot of numbers, and they'll just walk all over things and have a bit of nectar and walk on to somewhere else. So they're not maybe as specific, but in sheer weight of numbers they're good. And there's a grant that's just been got by a colleague of mine looking at trying to improve pollination of a number of difficult crops by rearing a number of fly species and releasing them for pollination. If anyone likes chocolate, then you've got to like flies, because the midge, a species of midge, a midge fly is the pollinator of chocolate. Okay, it has a very complex architecture in here. Here's the chocolate, the flower, and to get into that and pollinate it, you need this tiny little fly here. This is your chocolate after it's been pollinated. So there are certain regions where chocolate is from Central South America, generally associated with tropical rainforest sort of areas that these things are associated with. So you can't grow chocolate away from these areas, but the further and further they destroy some of these areas and try and put plantations in, the less movement of the midges you've got from the forested areas into the plantations where you are, and the less pollination. So they're getting really, really lousy pollination once they start destroying the adjacent areas that they need for chocolate. So, yep, without midge flies there'd be no chocolate. Okay, we also have a number of beneficials. We have adults as beneficials here. We have robber flies, which are predatory on the wing. You can see that's taken an aphid. And we have larvae. In this case, this is a hoverfly larvae, which are very significant predators of aphids again. So you can see them moving around the leaf surface here, taking there. So predatory ones. And we also have not as great an example as maybe we did with the Coleoptera or the Lepidoptera, but we do have examples of weed biocontrol agents using flies. Those that they use are typically the gallflawmers. We're not looking at the leaf miners, but the skeleton weed gall fly, St John's wort midge and the nodding thistle fly have all been introduced as part of usually a suite of insects to help control the weeds. There's not often just one insect involved. They target different parts of the weed. Biocontrol agents, fire ants. Just got 8 million in the budget last night from the federal government for fire ants. It's 13 million maybe it was for fire ant control. So right on the news last night, fire ants were winners in the budget, the newspapers declared. We showed you those last week in the lab. But there is a really fantastic family of flies, the Floridae, which are parasitic or parasitoids of fire ants, which have been used and looked at for biocontrol. You can see it homing in there on a fire ant. This family, Tachinidae, is a family almost exclusively, it is exclusively parasitoids. We talked about parasitoids in the Hymenoptera lecture. So these are fantastic biocontrol agents out in the environment. So there are quite a few good examples of parasitoids in the Diptera. Here is an example of one commercially being explored at the moment, the green vegetable bug. You can see it here with lots of eggs of this beautiful fly species with this amazing tibia here that's put eggs on that. So they're looking at that as a classical biocontrol program for green vegetable bug control. Okay, let's have a look at some examples of that I want to finish up on, which is mosquitoes in humans and blowflies in sheep. And I won't say a lot on blowflies in sheep because it's on the back of your handout, and I'll say various odd little bits and quips around mosquitoes in humans. But if we go back to that chart, we can see all those mosquito-borne diseases we've already mentioned there and went through from malaria right through to fluorosis to Zika to virus, etc. Okay, so here's a cartoon from my all-time favourite cartoonist, Larson. And in the book he wrote, he retired from cartooning many, many years ago. He just gave up. He had a book and he said this one caused the most concern amongst most of the people. He got lots more complaints about this than any other lecture about babies being taken down ant holes and things. What's wrong with this one? Who feeds on blood?
Use the shoes together.
Yeah. The blokes don't go out and feed on blood. It's the dad coming home there, but yeah, from that you can see it's got the tie on. So we're assuming that's male, and it's the females that actually do all the blood feeding, right? So it's not the males. They just flip around and look at flowers and nectar and things. Okay, so now let's have a look at malaria, from where malaria was to where malaria is today. And this is quite an interesting illustration to say that malaria was through a large part of the world. So through North America, through lots of Europe, across into parts of Asia that was never there, and through Northern Australia. This is the current band where malaria is. And a lot of this sort of malarial control has come through the use of insecticides and things to get diseases out of areas. And you can see obviously the more wealthy areas are those that have managed to achieve pushing out malaria from their different regions. Now let's having a look at here. Yes. Okay, so the Greeks had identified that this sort of disease, malaria, did exist, but it was the Romans who had one of the major issues, because the Romans tried to expand their empire right out through parts of Europe and into North Africa, and with them travelled malaria. Now malaria actually comes from the Roman mal air, or bad air, because they recognised there was an association between low-lying swampy areas and malaria. So they thought it was going to these areas and breathing it in. You got malaria, you got diseases. So that's where the original term came from. But it wasn't until sort of the 1870s through the 1900s when we really got decent levels of microscopes and other things, and there was a heap of Nobel Prizes in these sort of methods, where they actually managed to identify that mosquitoes are a vector and that the malaria actually is in blood cells. They all got Nobel Prizes and some of them died from their experiments in these sort of approaches. But now we know that malaria vectors and this life cycle of malaria is still a problem today. So let's have a look at what happened in the, around World War II in Italy, for instance. So here we have DDT. We were having 300,000 and 400,000 cases of malaria a year pre-DDT, and then when DDT was used, you can see it got down to less than hardly anything. So there were concerted campaigns through all sorts of areas to push malaria out of these areas, right through North America, Europe, etc. So that was very successful campaigns, despite what you might think about DDT in terms of its issues that go through to the environment and the transmission through to high water predators. It certainly had a large impact on disease incidence in a lot of different parts of the world. Now, I'll give you some practical advice rather than much more detail about malaria. And we have three different genera of mosquitoes here: Anopheles, Aedes and Culex. So Anopheles is the malaria vector, where the malaria vectors stem from. And if you're looking at larvae in a pond, the Anopheles reside horizontally onto the surface, whereas the Aedes and the Culex, and the Aedes transmit things like Zika virus, dengue virus, Barmah virus, a lot of the other ones. So we're really comparing Aedes to Anopheles here. They have a slightly shorter siphon than Culex. Nothing much in the pupae, but if you look at the adults, you can see a long palp in Anopheles and a short palp in females. But it's even better than that, because when you sit down and wait for a mosquito to feed on you, you can tell whether you're going to get malaria or dengue pretty well. So if you want to get malaria, you've got to get an Anopheles to bite you, and they sit at an angle that is like 45 or almost upright as they're feeding on you. Whereas if you're getting Aedes, so you're getting dengue or Zika, they sit more at a flatter sort of angle when they're feeding on you. So you have some idea of what disease you might get by what mosquitoes you let bite you. In terms of Australia, we have a lot of different species of mosquitoes, so it's not just a few. This is a chart trying to help with identifying locations and things of mosquitoes. And of course we've got Ross River virus and now Barmah forest virus here down in Tasmania. But interesting, if you look at a mosquito and you look at the wing of the mosquito, it has hairs along the wing, along the veins, which helps you identify that it indeed is a mosquito. Last one. Blowflies, Calophorids or Celia caprina, the sheep blowfly. Didn't get into Australia till the 1940s, 1950s, came in through Western Australia, came from South Africa. Okay, and then displaced local blowflies, which weren't nearly as bad as the sheep blowfly. Now we have issues, of course, with sheep blowflies. So this is the life cycle of sheep blowfly. This is the very last slide, I think, here. It is. So the life cycle is that they'll find a moist area here to lay their eggs on, where a wound is preferably, and then they will breed up on the sheep and then get into the sheep and feed on it and then pupate off it. So the problem we had with the breed of sheep that we brought into Australia and generally done merino is they have a lot of crinkles and folds on their backside, which is just a perfect moist environment for sheep blowfly. So of course what the initial response to that was to mules or cut the flesh off the back of here so you don't get sheep blowfly strike, which could be quite painful, but then that now has become out of favour for animal health reasons. So it still is an ongoing issue to try and better manage sheep blowfly than we currently do. And that handout there tells you a lot more about it. And with that, we have finished the entomology component of lower life forms.